She is an experienced clinical pharmacist with a demonstrated history of working in hospitals, clinics, academia, and pharmaceuticals industries. She is skilled in medicines optimization, patient education and counseling, medicines information, awareness and support, medicines use, and medicine safety. Strong clinical practice, teaching, and research professional with a master's in clinical pharmacy from the School of Pharmacy, University of London. With this brief introduction, I would like to request Dr. Mansi to take over the session.
Good morning, almost good afternoon to all of you present. After brilliant sessions that we've had on biosimilars, we'll make a head start and go into something that could be clinical after the point of part of the biosimilars life cycle. So we look at risk management and pharmacovigilance, which could be specific to biosimilars, and briefly before that, in general. So with a brief introduction to biosimilars and a risk management plan as pharmacovigilance included within that, and specific activities of risk management, we'll move on to good pharmacovigilance practices, including reporting and pharmacovigilance aspects, which are very specific to biosimilars, and then take us to risk minimization, which then I will follow on in the next session with actual case studies. So if we think about the current scenario globally, we have an aging population and significant number of conditions that are not simple to treat with standard therapies. And chronic life-threatening conditions do not help either. So we have these biologics in market. Since over 2 decades, we've had the biosimilars as well, so biologics before that. But the expense and the economics that are entailed within these present a limit to the access that is available for the healthcare system and patients in general. As it is end of patent life with multiple different biologics, we've had multiple biosimilar development processes and into the market. This has made them more affordable and accessible to individual patients and the healthcare system, and to more accessible in the sense of the number of patients who could be treated with these. If we look at the autoimmune rheumatologic diseases, it affects probably 1% of the Indian population. And mainstay of treatment continues to be conventional DMARDs with acute use of steroids in a variable manner. But biosimilars or biologics have been found to be significantly useful in aggressive disease or in case of severe disease, which is intolerant to use of standard DMARDs and immunosuppressants, or in case of poor relief as well as in case of side effects. TNF blockers, interleukin-6 antagonists, and some of the others have been used very widely in rheumatology. But the high cost has often led to discontinuation of treatment and a probable lethargy in use from the point of clinicians as well as patients. Although the intention is always to push for decent control in severe conditions and not necessarily good disease control in all patients, it still has been a challenge. So that has led the recommendations globally from EULAR and the American College of Rheumatology to prescribe that TNF blockers may be used as a start-off. And when these are not effective, people move to not necessarily a different TNF blocker, but to a biologic, which is a non-TNF DMARD. There are multiple reasons involved because of the inherent variability and the immunogenicity that biosimilars and biologics present. So this is just to give you a brief cap on what current scenario is in rheumatology that I largely practice in. There are multiple definitions for biosimilars, and if we look at the WHO, they are called as similar biotherapeutic products, or by others as follow-on protein products, subsequent entry biologics, and multiple different terminologies have been used globally. The overall intent is to prove similarity in terms of quality, safety, and efficacy to an already licensed reference product. And that is where biosimilars have entered the market. So in India, we have the CDSO describing it as those produced by genetically engineering techniques. And in the US FDA, they are termed highly similar to the reference product not necessarily identical, which is what we expect with biosimilars. And there could be some variation which is not going to be clinically therapeutically impacting the patient. As against this, in the European Medicines Agency has described these as products that show similarity to the reference product with reference to quality characteristics, biological activity, safety, and efficacy. And this is based on a comprehensive comparability exercise that is performed. So this is to give you an example of what the difference is, and I'm sure multiple others have done so. Monoclonal antibodies are the most complex forms of the biologicals that we know of, and there are multiple in use in variety of different conditions. So if we think of the kind of medicines we have, these are a diverse array of medicines that is with reference to biosimilars, and multiple different prophylactic vaccines as well as advanced therapy medicinal products are included in this group of medicines that we are discussing today with reference to risk. Related products are also defined as those that have a closely related active substance, but they are not necessarily authorized as biosimilars. And we have a few examples of that. So if we think about biotherapeutic products or biosimilars, these are relatively large complex protein entities which are very difficult to characterize, but with brilliant analytical techniques in place, this has been simplified. As yet, comparability exercises are not very robust necessarily, and that leads to a change in the risk management that happens. The uniqueness of these products is complicated by the manufacturing processes that they have and the inherent molecule on which from which they are derived and on which they are produced. All these factors make them sensitive with inherent variability because of the protein production or expression and the system and the inherent characteristics of production, as also due to the change in manufacturing processes which are certain to happen in the life cycle of a biosimilar in the market. And these could be multiple post-translation modifications which could eventually impact the clinical outcome and the therapeutic efficacy or safety or the lack of it. Important to understand that no biosimilar, we all know, is scientifically or technically identical to the originator or a different version of itself as well with a change in manufacturing process. This holds true even for different batches of the same product. The reason to understand this is that this is very critical in clinical practice because of the differences in production that would be there in different batches. Although we are aiming for consistency in quality and effectiveness, there would be some extent of difference in every batch. And thus, it is important that to the extent possible, robust manufacturing and in-process quality control procedures are in place. Going back in history, EU was the place where the first biologic was ever developed, and we've had some consensus, and EU has moved on to biosimilars after India. There has been a significant saving with the use of biologics and biosimilars in the healthcare system per se and in therapeutic management. Thus, obviously, we've had multiple different biosimilars being produced. If we look at the evolution of biosimilars, this is the kind of pattern it's followed. And alongside this has been a legislative evolution and overarching guidelines for the different molecules that have come into practice and different groups of medicines. To look at the Indian scenario, India has been in the biosimilar market since 2000. And this was with the launch of the Hep B vaccine. And then came on EU, which was the first in its biologic, but in Omnitrope was the first one to be licensed in Europe in 2006, followed by the US, which followed suit in 2015. Although there were a related product that was put into market in the US in 2007, it was not termed as a biosimilar. So that is what we call the related products. So this brings us to the point where this is a specialty, a therapeutic specialty, and thus also a specialty pharmacy area that we need to look into. India is needless to say a global player with a market since 2000, and the government and regulatory agencies providing enabling environment, still there exists a need to upgrade technology and improve manpower skill, which is the most important. The biosimilars market is not going anywhere because of the burgeoning population that we have, but also improvement in the manpower, the skill, and the clinicians that we have with reference to use of biologics and biosimilars. In clinicians, I do not mean just the prescribers in India. I also include clinical pharmacists and all the other health professionals who are part of this, including those from research and into development and manufacturing. The uniqueness of a biosimilars life cycle makes it important to understand the complete cycle from research up to the point where it is into the market and given to the patient. This is to create better confidence and acceptance of use of biosimilars in practice. Because of this, it is significant that there is a regulatory review throughout the product life cycle, which brings us to the regulatory landscape that we have with biosimilars. It is a complex landscape when we think about the development guidances as well as that for pharmacovigilance. And there is very significant diversity in what we have globally. So this is to give you an idea about the development of the biosimilar legislation and guidance across different countries and with the WHO, and this starting from 2000 or 2001 to the current scenario. So the biosimilars life cycle, if we think about the WHO and the development of standards, these are required to be implemented into the national regulatory requirements for biologics development and also for the pharmacovigilance and risk management. That is the primary aim of the WHO in developing these guidances. Obviously, there is going to be variability and not everything is going to be put up into the national requirements for development or risk management. The aim is overall to get a better convergence globally in the regulatory landscape as compared to what we have at present. So the Committee on Biological Standardization from WHO created different aspects of, worked on different aspects of this, which was the comparability and choice of the reference product, the key principles and concepts of licensing, and then the regulatory evaluation, which included everything from the development stages, the analytics, the safety and the quality, and then the preclinical and clinical evaluation. The strength and depth of requirement for each of these evaluations and the parameters are significantly different when we look at the originator as compared to the biosimilar, which is clearly depicted here. And there is even further change in the EU with more stress on analytical studies and results as compared to preclinical. And needless to add, the US and India will eventually move to in those lines. The most significant difference with biosimilars and biologics is that with biologics, we are trying to ascertain the clinical efficacy of the product. And with biosimilars, we are trying to demonstrate comparability to that reference biologic with reference to different areas like kinetics, dynamics, the safety and immunogenicity. And to the extent that minor differences between the two will not impact the final clinical outcome. So with a complex regulatory scenario that we have, the focus for approval largely relies on safety and efficacy data and knowledge from the reference biologic, similarity to it and extrapolation of data, as well as variability that is acceptable in the manufacturing process, which will again take us to practice and as to how that is going to impact risk management. So in India, we have had in the past guidances that have been created by a committee on genetic manipulation, and these were with the CDSCO till in 2012 and followed on with 2016. We had guidances on similar biologics, which clearly speaks about individual case-to-case evaluation of each particular application. This took us to the difference in the 2012 and 16 guidance with increased requirements in comparability testing that were required with reference to the reference drug and what was going to be the acceptable variability in terms of manufacturing to still maintain the same identity, the similar identity, purity, and potency to the reference product. So this guidance 2016 largely stressed on and with the aim to reduce residual risk with the use of a biosimilar, especially in terms of immunogenicity assessment, and thus became essential post-approval commitments, as in the sense of phase 4 studies and registries or biosimilar databases that had to be created in the process. So if we look at the need in pharmacovigilance, the post-marketing surveillance is a must, that is the phase 4 study, and the requirement at present is a minimum of 200 evaluable patients that are studied across a single arm study and within 2 years of marketing approval. In addition to this, it is important that at the point of marketing application, a phase 4 protocol is also submitted alongside to indicate the kind of evaluation that is done. But there is significant variability across the globe in the kind of post-marketing safety studies that are taken up. So in the EU, as an example, for in the UK, the additional monitoring requirements demand that these products are intensively monitored and thus marked with a black triangle into the first few years after marketing authorization. This is indicated and highlighted on the package insert, as also with an indication that this particular product is subject to additional monitoring. This is the case with all biosimilars that come into market and multiple other medications. This particular tabular version gives us an idea about the differences in development to market and actually reaching the patient as to what kind of evaluation and how many patients we've had and what kind of preclinical data we have, as well as the phase 3 study data, which takes us to post-authorization activities that are a requirement, as in the name of risk management plan and pharmacovigilance, that is to be included at the time of the marketing authorization application. This brings us as pharmacists and educators into the picture largely after the research, development, and manufacturing once again into practice. We have a significant role in creating awareness to improve informed prescribing with biosimilars and prescription monitoring of those prescribed biosimilars for those particular indications and patients. Access is a big issue, and market access is something I'll look into in the next session. This takes us to therapeutic monitoring with the use of biologics and biosimilars, and that is when risk management and pharmacovigilance in practice comes into being. So the risk management before, that is the pre-licensing, is different, and we're going to look at the one that follows. This demands a good understanding of pharmacovigilance, its rationale, and in association with biotherapeutics, which is what is the most significant aspect that we as practitioners and educators need to look at when we are training others and students. So as a classic example here, this is a scenario that we have had based on the WHO survey that was reported in May 2020, where we have 98 biosimilars that were approved in India as against 61 in the EU and 26 in the US. I'm sure there is a growing market, and these numbers would have changed, but these are the reports from last year. Once a biosimilar is marketed, it becomes an individual product, is important to understand, and thus it undergoes the same kind of post-marketing evaluation that would be required for a biologic or an originator. With biosimilars and the economics that we deal with, new therapeutic opportunities have grown in market and has improved the opportunity and options in clinical management. But with it comes significant challenges with reference to safety that includes the potential for immunogenicity, special considerations that are required for the pharmacovigilance, parameters that need to be considered for biosimilars and biologics, and thus have also come on overarching guidelines with reference to good pharmacovigilance practices that need to be looked at for development and pharmacovigilance of biosimilars. That takes us to the actual pharmacovigilance. So when we think about risk management, because there are multiple subsets of patients who are beyond the target population, and they may react to a particular biosimilar or a medication differently from others, that is the general population, that is being treated by a biosimilar. It is important that for us to understand that preclinical studies, pre-authorization clinical studies are insufficient with the small numbers that we have to extrapolate this to the larger population that would be eventually treated with biosimilars. A typical, and thus it becomes important to understand how to manage a particular risk once identified. But management of a single risk also in itself encompasses different steps, which includes the identification, assessment of it, and minimization measures that can be put into practice, and communication to the health professionals who are involved with it, with the use of a particular biosimilar, as also patients and carers who are directly in focus. Pharmacovigilance, obviously, in general, helps identify unknown ADRs or suspected ADRs and helps us assess risk benefit balance. And thus, over the years, we've had a brilliant pharmacovigilance system, which is quite robust in place for small molecules. But these aspects are not enough for the pharmacovigilance of biosimilars because of the complexity of the situation that cannot be gauged in the current market. For example, with again inherent and batch-specific manufacturing variability, immunogenicity, and traceability issues that we come across. The complexity of the scenario here is depicted in the EU as to the number of biologics and biosimilars as a brief example of what is in market and how much we are trying to manage in terms of safety analysis post-marketing. So if we look at this vast list that we have, it's important to take this into account with the WHO definition for pharmacovigilance, which speaks about science and activities relating to detection, assessment and understanding, and prevention of ADRs or any medication-related problems. But to be able to achieve this, good pharmacovigilance practices are required for reporting of all types of suspected ADRs and medication-related problems, and these should include medication errors as well as those issues that come up with overdoses or lack of efficacy issues that may come up. This is the complexity of just guidances that we have across 3 countries and continents. And probably in India, we just speak about the national guidance that we have as against the EU, where we have that across the EU, there are multiple different good pharmacovigilance practices. Even in India, the pharmacovigilance guidance that came out in 2018 may not be specific to biologics, biosimilars, but we have a guidance that was specific to vaccines. All of these put together with the global guidances that we have gives us an idea about how to manage risk better over a period of time when a biosimilar is in market. So thus, anticipation of issues that may be there due to safety, monitoring, identification, and the actual recording and reporting of any side effects or adverse events are crucial to improve treatments and optimize therapeutic outcomes. This takes us to an amalgamation of pharmacovigilance with reference to biosimilars. And once again, we have a complex regulatory landscape, which makes it essential that we look at a risk management system that could be in place. And it is essential for within a risk management plan that could happen that all medications, applications that are biosimilars have to undergo certain additional parameters. So if we look at a typical risk management plan, the EU risk management plan is one of the most widely used and the most robust at present. And this includes aspects on pharmacovigilance, risk minimization measures, with the aim to improve and monitor safety with use of biosimilars and to reduce risk. A typical risk management plan that we will look at will be the EU risk management plan. The template includes a product overview of the biosimilar that is being used, safety specification with reference to identified risks and potential risks, a summary of safety concerns. This in itself includes immunogenicity and associated clinical consequences with a fully evaluated, which needs to be fully evaluated and discussed as part of the initial marketing authorization application or a variation application. And this needs to be included in the summary of clinical safety of the marketing authorization application. In addition, for biosimilars, additional requirements for safety specifications are a must, and these need to be fulfilled for all biologics and biosimilars. Within the risk management plan is the pharmacovigilance plan, which includes routine pharmacovigilance activities and additional pharmacovigilance activity. These are required and should capture the overall safety, quality, efficacy profile determinants and the complex manufacturing processes with inherent variability and batch-specific variability, as also the different steps that are there in the manufacturing processes, which could be upstream or downstream, and the immunogenicity, which could come up as a result of the kind of manufacturing process that these products undergo, which takes us to post-authorization safety studies, which are a requirement as well. With the post-authorization safety studies come up risk minimization measures that have to be taken up, post-marketing authorization, and these will normally lead to an update to the risk management plan, which could be due to multiple different reasons. It could be an update to the risk management plan of the reference product. It could be due to manufacturing changes or a potential impact of the manufacturing changes. These in turn demand risk analysis and an update of the risk management plan, because this will or could impact significantly prescribing pattern and prescribing information that needs to be put into place for that particular biosimilar. Within the pharmacovigilance activities in the risk management, we have different routine practices that need to be taken up, which is managing and reporting of adverse reactions. Within management is included identification, evaluation, with multiple risks that are present and multiple medications that a patient may be on. So that takes us to individual and suspected ADR reporting, as also aggregate reporting, as in the case of periodic safety update reports. These will present estimated exposure and usage patterns of biosimilars and the risk and safety that is reported as a result. It should also give us an overview of the signals, which could be new, ongoing, or close signals, which again support risk evaluation that needs to be put into place as a continuous pharmacovigilance activity, which will be included as part of signal management and additional monitoring parameters. When all of these aspects are taken into consideration, unless the safety or lack of effectiveness or efficacy is actually communicated to health professionals who are into prescribing and monitoring, or to patients, the earlier activities do not hold any strength. So here it is important to understand that, like for all medications, there are different sources and types of adverse reactions, and it's important to understand where a reported ADR has its source. So when we speak about the predictable pharmacological ADRs, in case of biosimilars, these could be things like epoetins showing higher hemoglobin levels than what our target value was, or there could be unpredictable pharmacological effects and immunogenic properties, which could be due to the active substance itself, where there could be an exaggerated or diminished immunosuppression that we may see, or an immunomodulation with a particular biosimilar, or some immunogenic property that is being introduced because of the excipients in addition to the active substance. There could be process-related impurities like the host cell proteins, or the host or disease-related susceptibility, which could be due to the immune system, autoimmunity, or production of auto antibodies and anti-drug antibodies, as well as inflammatory parameters that come into effect with use of biosimilars. A risk-benefit assessment and specific considerations are absolutely essential to make the pharmacovigilance capabilities more robust and product-specific. And this once again takes us to good reporting practices. So within good pharmacovigilance practices, we will have good reporting practices. So in spite of good pharmacovigilance systems in place in multiple countries like in the EU and the US and Australia or Netherlands, we have seen with significant misattribution and a lack of appropriate causality assessment with, for example, in case of filgrastim, there was a significant difference in from 50 to 90% in misattribution of the particular ADR. So these are significant challenges for biosimilars in addition to the standard ADR reporting challenges that we already have as to who has to report, what can they report, where, the kind of reporting support that we should be having. And the biggest issue is what happens to the data and traceability of the actual ADR that we have. That takes me to give an example that significant work has been done with all marketing authorization holders that they have on their websites as also on the competent authority websites. Different ways and information on who can report and what can be reported by health professionals and non-health professionals or patients. So with a robust pharmacovigilance system in place, the most important is that the marketing authorization holder and regulatory authority work together to enable urgent action which may be required in case of specific reporting that is done with reference to biosimilars ADRs that may be batch specific and could be affecting more than one particular individual. So when we look at good reporting practice, a competent authority should ensure and have appropriate measures in place to identify biosimilars with reference to traceability and ADRs with at different points of prescription, dispensing, and where they are actually sold or administered. Thus, the marketing authorization holder with competent authority need to have appropriate records in place. This demands that there is brand and product name and batch number reporting in every ADR form that is being submitted, which helps traceability with individual reports or even helps in cases when there is more than one ADR reported for a particular batch and takes us back to issues that could be traced to a particular batch rather than the product itself or as a whole. With reference to ADRs, it's also important that at the national level or individually at the marketing authorization holder level, clinical complaints are largely dealt with by medical personnel in addition to other clinicians and those involved in pharmacovigilance activity at the marketing authorization holder side. Issues about quality complaints should ideally also be dealt only by the quality personnel in addition to the clinicians who take this matter forth. Source of report will also drive the kind of information that is being given, and thus there should be different reporting systems and forms that make it simple and accessible for health professionals to report as against the patients and carers. The marketing authorization holder and the competent authority should encourage healthcare professionals to provide patients with all information on a particular product and batch that they are being given at whichever the point of access to that particular medication is, or at the point of administration. But in spite of this, there need to be follow-up procedures in place to obtain batch numbers if they are missed at the point of initial reporting. In addition to this, it is good practice and good to have information on prior administration, that is administration history of biosimilars or biologics, which will give us an idea about collecting information on defined clinical outcomes to aid us in risk evaluation in case of immunogenic reactions or lack of efficacy issues, which will enable thorough evaluation and causality assessment with use of biologics because of the inherent chances that they have in the development of neutralizing antibodies and immunogenicity. Marketing authorization holders as a school of thought also have the need and the demand for developing and detection parameters for anti-drug antibodies that may have come up in a patient's blood sample. But whether to make these requirements and blood collection and testing mandatory is a different area to look into. And where do we report is the next aspect of ADR management and reporting. So marketing authorization holders and competent authorities could have within their websites information and adverse reaction reporting forms or an opportunity to directly communicate with these authorities to enable reporting of an ADR. These forms or direct communication activities should be able to communicate and facilitate appropriate capture of all essential information with reference to the demographics and traceability variables, which include once again the batch and the product information. Classic example here is what happens at the marketing authorization holder's website, the kind of adverse event reporting opportunity that is present. And this includes information on what can be reported and who can report it. In addition, there is also scope and opportunity to look at areas and aspects that a health professional can report and as against what a non-health professional or a patient could be reporting. This is slightly different from the US system where the forms, again, online forms are used, but the reporting is directly to the National Pharmacovigilance system in the US. So in India, we have a slightly different variable in the sense if it is reported to the marketing authorization holder, which could also be the case in the other country, in some other countries, this is then taken on to reporting as we see in the case of the Pharmacovigilance Program of India, where we could report. And this is a standard form we have for healthcare professionals. These are then captured and reported to the international body, which is for the international drug monitoring that we have. So as the National Coordination Center by the Indian Pharmacopoeia Commission, we have reporting forms on their website and those for individual patients as well in different languages. In addition, we have reporting support and helpline numbers that are managed at their level at the National Center for reporting and for general public to be able to report. These are in addition, these move on to the Uppsala Monitoring Center, which is the WHO collaborating center, as we all know. And India has been, the Indian Pharmacopoeia Commission is a full member since 1998. The significance of understanding this is that this is a global effort to identify and reduce harm or risk from therapy, as we all know. And information flow is through a Vigi-based system, which is helps capture reports of suspected ADRs and in individual case reports from multiple different member states. And there is a repository of over 20 million reports, which was by practically 2 years back. This helps us at the WHO and through the International Drug Monitoring System analyze global patterns of suspected harm with use of all medications, and this includes biosimilars as well. So on behalf of the WHO, the UMC presents its research and operational support, technical support, and guidance to national centers. And that is why I have put this into picture here, because the same kind of system as the Pharmacovigilance Program of India and the Indian Pharmacopoeia Commission and the National Collaborating Center has from the UMC as a full member of the International Drug Monitoring program that we are part of. This helps develop, this program also helps develop and supports countries in reporting and data management tools that would be required at their level. And VigiFlow integrates the international standards to record and manage individual case reports at the national centers as well. And obviously then there are training and publications from the international center. The reason to look at this is because of the good pharmacovigilance practice that we have reported and in the Indian document as well with the pharmacovigilance guidelines in 2018. There has been significant improvement in the amount of information people have and prescribing and reporting practice that we have seen with biosimilars and other medications, but we have a long way to go. This document in India also requires us to have a risk management plan in place to identify and characterize, or in addition to minimizing risks through a product's life cycle, and ensure that there is a balance between the benefit and risk. With the increasing awareness, changes have happened, but tighter monitoring is required with biosimilars and biologics to ensure appropriate efficacy and safety. If we look at the EU regulations as against this with good practice, It is mandatory to have in every individual case report as well, a brand name and batch traceability. These are called as, in addition to these individual case reports, are the periodic benefit risk evaluation reports, as they are called in the EU. These are submissions as per the EU reference date list, and these are these present and provide information on the frequency of the PBRER submissions that are required, the data log points that may be present, and the submission date, the requirements for submission of these PBRERs for the different products. As against this in the US, these are called as the periodic adverse drug experience reports, and the requirement is submission on a quarterly basis for the first 3 years and annually thereafter. As against this, when we look in the pharmacovigilance document we have in India, we have the periodic safety update reports that are required as 6 over a period of 4 years to help identify new safety signals and determine the benefit and risk changes over a period of time, as also risk communication, which is an essential part for regulatory authorities to and acts as a needs indicator for the risk management initiatives that have been taken up and to track the effectiveness of these initiatives that have been put into place. So if we look at the requirement of a periodic safety update report, these are critical because of the limited exposure in the clinical studies that are done pre-marketing authorization. Each PSUR should include all dosage forms and presentations and strengths of a particular biosimilar, and every single PSUR undergoes a particular cycle over a period of 4 years with 6 submissions. But beyond this, with any change in the strength or dosage form which has been included in the initial PSUR, a change in the manufacturing process, or a change that may have been inherent to a biosimilar, the cycle restarts and follows on for another 4 years. Safety issues are thus required to be individually monitored as part of the regulatory authority requirements, and these may demand an extension of a particular PSUR. This is to get an estimated exposure and use pattern and to support signal management of management processes that are put in place by the marketing authorization holder. And it is important at this point that there is actual usage data as against the sales data that is present. So this is where the source of information, what we have from clinics and practices, is very important. Data sources will decide as to what the actual usage data is and also the strength of the information that we have. The overview of signals and the type of signals we have will also aid in risk evaluation for these particular reports that we have. A classic example I have here is that of a safety signal that was for teriparatide and the risk of shivering that was reported by the Saudi Food and Drug Authority. It gives us an idea of the kind of individual case safety reports that are present globally, and as to how the WHO UMC has a causality assessment, which does probably evaluate in a way that some of these reports were identified as having a probable or a possible correlation to teriparatide. But the significance of this was not enough to correlate it completely to that. So the information here that we got here was again from the database that was the Vigibase and all the ADR reporting that has happened globally. This is to give an example as to what kind of global the Vigibase works on and the kind of individual case safety reports that would be useful in the long run. So this demands, signal management demands a continuous pharmacovigilance process in place, which should be sensitive enough with processes that are inbuilt to enable early identification of biologics and biosimilar variability based on theoretical risk assessment and monitoring that we would have, and which can actually arise in practice during any point in the drug life cycle, so the biologic life cycle, that is including up to the point it remains in market. This will also help detect new clinical risk and safety or efficacy parameters that emerge anytime during the life cycle. And important is at this point is the post-authorization exposure information and the clinical setting and the significance of the clinical setting. Because we are looking at the actual product specific usage data with different sources and methods of procuring data. Important is that we have reliable and updated information to be able to analyze the denominator data of suspected ADRs and support continuous signal detection on an ongoing basis to evaluate trends and find trends in reporting rates based on new signals or identify new signals that can eventually come up. And the significance of this in terms of clinical outcome is what we need to assess and evaluate. So signal detection needs to be, for that reason, specific to the product and to the active substance of the product. And it should be performed at the level of the product name and the active substance itself. And this demands a root cause analysis based on every batch. This will help get data specific to evaluate, that is the batch number, the code of the delivered sold batches and the size of the batch, where it has been delivered, and what is the population, the target population that we need to look into to bring back traceability and what action needs to be taken in case of an ADR that is suspected. This will help strengthen the processes for routine pharmacovigilance and aid in early detection of new risks that could present with changes in product quality, safety, and present over a particular period of time and not necessarily immediately. Beyond the routine practices, we have additional monitoring, which has become a requirement since 2011. So all biosimilars are subject to additional monitoring as per the regulations. And after 5 years, unless requirements are added on or there is a period of additional monitoring that is extended by the regulators, or there is a change in the product or the process, at the end of the 5 years, if the safety parameters are met, the biologic or the biosimilar is taken off the requirement for additional monitoring. So this is like we speak about the black triangle drug, a black triangle that we have, that we spoke about. So that takes us to safety communication. All of this information that we gather with reference to risk management and pharmacovigilance needs to be actually communicated, but the complexity of biosimilars and biologics as added on with the complexity of the manufacturing process and inherent and that specific variability that we see, as also the risk which will change over time, and ADRs or complex effects that are due to immunogenicity. It is important that we address specific aspects that are in a manner and communicated directly or specific to a product in a manner that is appropriate for patients as well as the general public and to health professionals who may not be very well versed with the use of biosimilars. Communicating risks of biosimilars and the complexity of it needs to take into account the appropriateness and adequacy of the language and information that we provide and that is presented, which may vary between the different groups, target groups that we have to communicate the safety and the issues with safety are. But at the same time, to improve prescribing and use of biosimilars, it's important that we build confidence with the use of these, and this should be through adequate information sharing, which again covers the complete life cycle of the biosimilar, from manufacturing to the point where the biosimilar is actually administered to the patient and monitoring thereafter. We need to overcome beliefs and myths with reference to use of biosimilars to promote better prescribing and use of biosimilars. And the safety mechanisms and communication that needs to be done should also be with reference to improving safety surveillance mechanisms to encourage appropriate and reporting per se and appropriate reporting for potential for ADRs and biosimilars. It should include traceability issues which need to be communicated to the individuals who we are speaking to, because unless there is traceability, it is very difficult because of the batch-to-batch variation to be able to actually communicate the kind of risk that would be not necessarily inherent to the product per large, but to a particular batch. This demands appropriate evaluation on storage and handling and the cold chain requirements, with temperature as the most important environmental factor affecting the quality and stability of a biosimilar. That takes us to the devices itself, which are in direct contact for administration. Safety communication should, in general, based on all these aspects, communicate efficacy and extrapolatability from the reference, the comparability and interchangeability of the molecules that are in market. There are different documents to be able to get significant information on this, which can be used as reference documents. So at this point, we will look at the most important aspects of biosimilars that affect the pharmacovigilance and what we are monitoring. This will include immunogenicity, stability and cold chain, and product traceability after the manufacturing variability that we've discussed briefly already. So when we speak about immunogenicity, this is introduced or could be altered post-authorization at any point in the life cycle of a biosimilar in market or in development. There is potential that this has a potential to alter the safety and efficacy of a particular product and the effectiveness with clinical consequences that we will see in therapeutic management. The immunogenicity evaluation requires that there is, during the product development and prior to authorization of biotechnological products, there are specific guidance and these should be followed, and post authorization as well, there is significant evaluation and assessment that is required with reference to immunogenicity. The most critical challenges that we see could be product-related, antibody assay-related, or treatment-related with reference to the sources of immunogenicity or the factors that influence immunogenicity. These include use of non-clinical models and analytical methods or bioassays, which may not be appropriate to predict the immunogenicity parameters in humans with that particular biosimilar. In addition to this, the pre-authorization clinical studies have limited sample size, and the rarity of certain diseases may not allow rare consequences to be captured pre-authorization. And thus, there is need to incorporate immunogenicity accountability in its totality in any risk management plan, and to the extent that can be gathered prior to marketing authorization. The different factors or sources of biologics and biosimilars immunogenicity are multifactorial and involve one or more product-related process factors, that is, could be the choice of cell lines, post-translation changes that can occur, as also alterations to the structure within the process, that is downstream, during downstream processes, impurities that can come up, or the choice of product, device, or container that is used. There could be treatment-related factors that can influence the immunogenicity, which would be a dosage regime that is being followed, or specifically to a particular frequency or duration of use of a particular biosimilar, and the tendency to form anti-drug antibodies in the process. In addition, there are patient factors, which would be like the genetic background of the particular patient, the strength of the immune system, the immune status per se, if not the strength, age, and the use of different medications called concomitantly, which will obviously introduce a factor of multiple risk and causality assessment challenges. But at the same time, the clinical consequences of immunogenicity could lead to a partial or a complete loss of efficacy due to formation of neutralizing antibodies, or could have an altered pharmacokinetics due to antibody binding. But general immune effects like anaphylaxis or formation of immune complexes and introduction of cross-reactivity within endogenous proteins or other autoantibodies is also not unknown. Classic example here is I have of immunogenicity that was reported with infliximab and development of anti-drug antibodies in a certain number of patients, which was 5 to 12% were higher in the biosimilar cohort, was reported at individual time points of determination. But at the same time, it's important to understand that these could be directly correlated, but will it impact clinical use of a particular biosimilar or not is the kind of compatibility weighing risk-benefit analysis that we need to look into. So the clinical consequences could be serious and life-threatening with reference to immunogenicity, but do not necessarily always have to be. In certain situations, we may only see transient appearance of antibodies or a change in the risk-benefit balance, which may not necessarily impact clinical outcomes or the therapeutic use of a particular biosimilar. So it's important to understand that these are inherent to all biosimilars, and we need to look at it in totality. The variability that is introduced from multiple sources and risks, including manufacturing, takes us to the point where biosimilars are actually administered to the patient. And efficient and safe biosimilar devices for delivery require seamless transportation and storage system. These are mandatory factors for use and transportation of biosimilars and up to the point of administration in the patient. Quite a few biosimilars, multiple ones are for parenteral delivery, and the kind of device that is used may be the primary container of the active drug, which is directly used for administration. Because patent requirement is that biosimilars device is not the same, cannot be the same as the innovator device. With every biosimilar that comes into market, this is an aspect that needs to be looked at in complete as against comparability that can be done in other aspects with the reference product. As the active substance is directly delivered by the device in which it is contact with all the time, from the point of manufacture and into market to the point of administration. This device and the container plays a significant role in the quality and the volume of the product that is being administered. And it is important that there are efficient regulatory parameters in place to ensure safety of devices in addition to the molecule, the biosimilar itself. While we are looking at these parameters, the precision of dosing is very important with biosimilars or with any other medications, which may be slightly variable with standard medications and slight differences in the adult age group, which do not have comorbidities. But that is not necessarily overcome and cannot be overlooked in case of biosimilars. But in addition to the kind of device which will appropriately deliver the right and quality and safety parameters fulfilled medication to the patient, is also required that these devices are easy to use and comfortable and convenient, because that will directly impact patient adherence. Essential to this is that a marketing authorization has strict process controls in place for biosimilars and biologics to ensure that manufacturing processes and standards remain within authorized specification. But beyond manufacturing and release, the overall product stability is maintained by adherence to appropriate storage and handling conditions, cold chain and good distribution practices that need to be in place. And these are very important post-manufacture and release, and these need to be incorporated as part of the evaluation in the risk management plan. Temperature is one of the most important factors, that environmental factors that affects a biosimilar quality and safety. So it is important that temperature regulation is appropriate during the transport and storage of the biosimilar, which will help maintain to the extent that it would otherwise impact the efficacy and safety of the particular product. Thus, vehicular transportation, refrigeration at different points, and storage within the pharmacy are factors that are very critical to safe delivery of the biosimilar to the particular patient. Often, these parameters are overlooked till there is an actual report of an issue with quality of a particular product or lack of effectiveness. So it is important that these parameters are included in the pharmacovigilance and risk management plan for any biosimilar. Non-adherence to these processes and standards could affect the stability or the quality of the biologic and the biosimilar, which in turn may affect or alter or introduce immunogenicity or contamination in the product. Although very rare, but it is feasible for the product already that is released and in market to show such defects and deviations. And these could be specific to a particular batch if there is batch specific release that is being followed in particular territories and which can be traced back to the manufacturer and the patient per se. Life cycle pharmacovigilance is at the level of the products and batches is thus very important. This takes us to product traceability, which is one of the key requirements for pharmacovigilance of biologics and biosimilars. There is the need to ensure that there is a continuous product and batch specific traceability in place when a product is in market and in clinical use. To be able to identify and assess post-authorization manufacturing variability over time, which needs to be done within and across products with similar active substances, or which could be specific to a planned change in a manufacturing process, which demands comparability to be reassigned to the originator after the manufacturing change, which is different from establishing biosimilarity in the first place. The minor structural changes may result in rare or delayed safety events that may not be detected until after marketing authorization because of the lack of the larger number of patient exposure that we have in the pre-authorization clinical studies. It is important that post-approval manufacturing changes that occur for all biosimilars, that such changes, when they occur, have an appropriate system in place to ensure maintaining product quality, safety, and supply. Or there has to be a system to be able to identify these. It is crucial that supply can be traced back to different patients and locations, and these could be based on the point of administration, point of dispensing of biosimilar, and it has to be essential that this is also across products with the same INN that need to be readily distinguishable in order for new and emerging product specific or batch specific safety concerns that are reported in the process. With any change that happens in as a result of inherent variability or a batch specific or a manufacturing variability, essential consideration is thus always it takes us back to batch traceability, and thus the product name and batch specifics need to be clearly indicated to and shared with the patient at the time of administration or supply of a particular biosimilar, because biosimilars are of diverse variety. They are diverse in nature and they cover a wide array of diseases that they are treating. There are multiple products and therapeutic areas as well as varied clinical settings in which they are used. Integration of this parameter of traceability with infrastructure is critical. So if we look at hospitals, as an example, because of the diverse array of products that for a wide range of therapeutic areas, traceability needs to be fully integrated into different healthcare settings. Most products are supplied in a hospital setting, and there should be record linkage to be able to trace these back. In scenarios where this is not possible, there should be routine barcode scanning at all points in the supply chain that is present. National health authorities should be able to support better integration and automation of prescription information. How realistic this is for the national authorities to do is going to be once again based on the territory, the country that we are looking at. The reason this is of significance is because these may influence significantly and drive prescribing practice, or positively or negatively, and this could also have a significant impact on product interchangeability or switching between different molecules, different biosimilars and biologics, and whether that is permitted or not. Thus, traceability is one of the most significant parameters in association with all the other biosimilars parameters that we look at. Here we have an example of the different biosimilars like Humira, MabThera, Remicade, and Forteo. My point here is that in all of these situations in practice, we have seen that when there is need to change from the biologic, that is the innovator, to the biosimilar, there is a significant amount of pharmacovigilance, risk management, and prescribing involvement that is required across different individuals, across different settings. And I will speak about these in greater detail with examples in the next session. But it's important for us to be able to communicate the patient the manufacturer's information, standard acute management that we would have in case of a medication related problem, and what evidence we have. Thus, these are to be initially communicated to the competent authority and appropriately to a particular patient at the point of a switch or a shift to a biosimilar, or in case of initiation of a biosimilar or an innovator product. In the different healthcare settings that we have, product name and batch number of the administered biosimilars need to be recorded by the health professional and provided to the patient, as I've just mentioned. This is a must because different versions of the same active substance could be on market and could be interchangeably used by the same patient. Whether this is permitted or not is a different matter. The pharmacovigilance of biologics and biosimilars is further complicated by the potential for delayed immunologic reactions that happen with the use of biosimilars and these products or any biotherapeutic products, or a lag between the administration and the appearance of a serious reaction as can happen with neutralizing drug antibodies reactions. Or in situations where there is a switch between related biologics, the time lag and the challenge that happens may lead to attribution or inappropriate attribution of a particular medication related problem or an ADR to that particular biologic or biosimilar. Experience with use and demonstration helps traceability to ensure that these are attributed, any adverse reaction or a medication related problem is attributed to the right product and to the right batch. The other big challenge we have with biosimilars pharmacovigilance is that it's very unlikely that a particular patient, or we could say no patient is likely to be only on a biosimilar or a biologic. There are most likely going to be multiple standard treatments, comorbidities, and management of other conditions, chronic conditions, or acutely, which may hamper causality assessment in case of individual case reports or in case of suspected ADRs, and comparison of frequency and severity of the known ADRs of the reference product with that of the biosimilar becomes important at that point to detect possible differences between the two products or a change in the causality that could be presented in the process. Trends in ADRs that could be reported with specific batches should be calculated because these may help identify an issue that could be specific to a particular batch only. The other classic issue we have with biologics and biosimilars is the risk that can develop over time. This is inherent to structural changes in the molecule itself, or it could be derived from the cell line that is being used for the development of that particular biosimilar. A change in the risk-benefit profile may over time post-approval through expanded use and patient characteristics, larger population exposure, demands that there is a continuous pharmacovigilance system in place till a particular biosimilar is in market. As an example, there are different groups that are made and there are different committees that are designated to capture such information and support signal management appropriately. In the EU, there is a Pharmacovigilance Risk Assessment Committee with different individuals and roles being designated to different groups within the committee. And there is reporting through EudraVigilance, which captures data of ADR reporting and pharmacovigilance data from all over EU. This presents opportunity for additional improvement in the system because not all member states report to the extent that is required and appropriately. This is an example. So then the Pharmacovigilance Risk Assessment Committee, the National Competent Authority, and the EU marketing authorization holders will then work with the individual member states for better reporting practices. If we look at the routine pharmacovigilance activities that are part of a risk management plan, these again take us back to traceability with variables of traceability as in batch recording and reporting, which will impact the clinical setting and the impact of the clinical setting on that particular reporting, and whether there are measures that are routinely in place to follow case reports that are identified and reported. The signal detection activity should be able to correlate to specific safety issues which are specific to particular batches and across batches specific to a particular product, to an innovator, or to the biosimilar. Identification of potential risks is as important as in the case of adverse events of special interest, and this demands specific safety surveillance that has to be done post-marketing for every biosimilar. The additional activities in pharmacovigilance that are required is that the marketing authorization holder, in collaboration with national competent authorities, should work towards improving traceability and measure background adverse event of special interest rates and monitor this as a continuous process through development of different registries and data banks, as well as incorporate safety monitoring that would be related to the reference or the concerned product and which may change over time based on the risk management plan updates of the reference, which will impact that of the biosimilar as well. The need for these updates happens because of manufacturing changes, immunogenicity, post-authorization safety studies, and important thing is that whether this is going to be clinically relevant needs to be accounted for as part of the risk management plan update that we submit as a follow-on from the initial RMP submission at the time of application for the marketing authorization. The need for a risk management plan update also demands emphasis on batch-specific pharmacovigilance and to be able to distinguish pre and post change, which could be any change in the product, and whether the risk was incorporated because of a change or simply because of the greater exposure to more number of patients, and that is what helped identify the risk. Thus, these risk management plan updates clearly indicate safety specification of a proper pharmacovigilance plan and risk minimization measures that need to be put in place. The classic example of these measures are to be able to work across different settings for a particular product use and to improve traceability, taking us back to the same areas that are part of the pharmacovigilance practices in signal detection and identification of important and potential risks that are as part of the safety surveillance. A marketing authorization holder, in addition, also is required to do regular literature searches for identification of individual case reports of safety and measures that need to be introduced to routinely follow these case reports. A review of reporting in social media and newspapers or other public media is also something that is gaining attention, because there will be situations when people and patients may not want to report or not have the right support or information to be able to report to a marketing authorization holder or a competent authority, but would want it as public information that a product is safe or unsafe to use. Example is a literature review here as was recommended for teriparatide, where it was reported that there is a risk of shivering with the use of teriparatide. Literature review has to be conducted by the marketing authorization holder on a regular basis to identify any additional risk that has been reported globally. And this is an example of what kind of parameters would be looked at. In addition to the known risk is the new clinical risk evaluation. Once identified, root cause analysis is essential to evaluate the ability to minimize that particular risk through use of different analytical studies and bioassays or different practices that are put into place at the level of development or in practice. And to improve traceability, the maximum number of risk minimization measures that need to be put into place are provision of information to the patient or as inclusion in the patient file and documentation, information about the batch number, product name, and the summary of product characteristics that was in use at that particular time when the product was in market and administered to a patient. The patient package information or the product information that is given to the patient. In addition, it is important that this is all used with relevant educational material with appropriate language and directly communicated either to the patient or a health professional, and any promotional material that is used has to be in line with the requirements for a kind of communication to minimize risk. In addition to this, to improve traceability, there could be other tools that are used for, in addition to the packaging parameters itself, to manage better the record keeping for patient files and sticky tear-off labels. If you see for pediatric vaccinations, we often see that there are additional labels on the packaging itself, which need to be taken off and put onto the patient file to indicate as to which product, which vaccine, prophylactic vaccine was given to that particular child at which point in time. Obviously, we don't follow that in case of COVID and the different vaccines that we have with that. That takes us to understanding where we as pharmacists and educators stand with relevance to safety and risk management and pharmacovigilance, which I will look at in detail in the next session. And I'd like to thank you all for hearing me out.
Yeah, thank you, Dr. Mansi, for your detailed presentation on the various aspects of pharmacovigilance and risk management relating to biosimilars. And I request audience, if you have any questions, please post on the question box. We're not getting any questions.
Yeah.
If any more, you can send by email also, that may be responded. And with this, I thank Dr. Mansi for your valuable time and accepting our invitation to deliver this excellent talk. And with this, I close this session, and we'll meet again at 2:30 for the final scientific session of the final day.
Thank you to all present, and thank you to the organizers. Thanks, Dr. Sundaram.